Henry Nottage Moseley, 1880. Naturalist aboard HMS Challenger, the expedition that basically invented oceanography. He pulled creatures from the deep Atlantic and wrote this: "Other animals attain under them gigantic proportions. It is specially certain crustacea which exhibit this peculiarity." He was describing a sea spider the size of a dinner plate, a pillbug the length of his forearm, animals that had no business being that large, and yet there they were, hauled up from a place no human had ever seen. Moseley had discovered a pattern. He just couldn't explain it. 145 years later, neither can we. Deep-sea gigantism. The tendency for creatures living below about 1,000 meters to grow dramatically larger than their shallow water relatives. The pattern is everywhere. Giant isopods, armored crustaceans related to the woodlice in your garden, except these ones grow to 0.5 meter. Japanese spider crabs with leg spans of nearly 4 meters. Oarfish stretching to 7 meters and beyond. Giant amphipods, giant sea spiders, giant squid. The deep ocean takes familiar body plans and scales them up like someone adjusted a slider and forgot to stop. The fact is obvious. The explanation is a mess. There are 4 main theories. They've been competing since the late 1800s, and none of them has won. Each one explains part of the pattern. Each one breaks when you push it too far, and the disagreement between them tells you more about the ocean than any single answer could. Theory 1. Bergmann's rule. Proposed by Bergmann in 1847, originally for warm-blooded animals. The idea: in colder environments, larger bodies lose heat more slowly because their surface area to volume ratio is lower. A bigger animal keeps its internal temperature more stable. Makes sense for mammals. Polar bears are larger than sun bears. Makes some sense for the ocean. It's about 4 degrees Celsius below 600 meters year-round. Cold water, big animals. Neat. Except for one problem. Bergmann's rule was designed for endotherms, animals that regulate their own temperature. Most deep-sea giants are invertebrates. They don't generate internal heat. The math still works on paper. Lower surface area to volume ratios reduce metabolic cost even in ectotherms, but the mechanism is different. It's not about retaining warmth. It's about spending less energy per unit of body mass. The rule applies, but for the wrong reason. Theory 2. Kleiber's rule. Max Kleiber, 1932. The bigger an animal is, the more metabolically efficient it becomes. A whale uses less energy per kilogram than a shrimp. In the deep ocean, where food falls from above as marine snow, a slow, sparse rain of dead organic matter, efficiency isn't a bonus, it's a survival requirement. A large body stores more reserves, burns them slower, and can wait longer between meals. The giant isopod Bathynomus giganteus demonstrates this with uncomfortable clarity. One specimen at an aquarium in Toba, Japan, refused food for over 5 years before dying. 5 years. No meal. Its stomach can expand to fill roughly 2/3 of its body cavity when it does eat. And when it doesn't eat, its fat reserves sustain it at a metabolic rate so low it barely registers. Classic tank build. Maximum armor, minimum upkeep. The deep sea rewards patience the way the surface rewards speed. Theory 3. The island rule. Originally proposed for actual islands, isolated ecosystems with small animals tend to evolve larger, and large animals tend to evolve smaller. Think of it this way. On a real island, a mouse has fewer predators, fewer competitors, and the same food base. Over generations, the mouse gets bigger. The deep sea operates like the largest island on Earth. except the island is three-dimensional, pitch black, and roughly the size of all the continents combined. Isolated, resource limited, low predator density, low competition. Under these conditions, the selection pressure that normally keeps small animals small, predation, relaxes. And the pressure that rewards size, intraspecific competition, energy storage, mating advantage, takes over. The result looks like gigantism, but it's really just natural selection with fewer constraints. The island rule is elegant. It's also frustratingly hard to test, because you can't build a control group for the largest habitat on the planet. Theory 4. The one that changed the conversation. Chappell and Peck, 1999. Published in Nature. They analyzed body length data for over 1,800 species of amphipod crustaceans from 12 sites worldwide: polar, tropical, marine, freshwater. Their finding, the maximum potential size of these animals, correlate directly with dissolved oxygen availability. Cold water holds more oxygen. More oxygen means cells can grow larger. Larger cells mean larger organisms. And below about 1,000 meters, where temperatures stabilize near 4 degrees, oxygen-rich water circulates through deep thermohaline currents, creating what amounts to a chemical permission slip for size. The oxygen temperature hypothesis was the most elegant model anyone had proposed. It had data, it had mechanism, it had geographic consistency across 12 independent sites. For context, that's like running the same experiment on every continent and getting the same answer. It should have settled the debate. It didn't. In 2019, researchers tested the hypothesis on Antarctic sea spiders, pycnogonids, animals that breathe directly through their exoskeleton. No lungs, no gills. Oxygen diffuses straight through the chitin. If oxygen limits size, then larger sea spiders should perform worse under low oxygen conditions. They didn't. Large and small pycnogonids showed no significant difference in performance under hypoxia. The oxygen hypothesis predicted a penalty for size. The data didn't deliver one. And there's a deeper problem. Some genuinely giant organisms live in warm, oxygen-poor water. That shouldn't happen if oxygen availability is the ceiling. But it does. The model works beautifully for amphipods and gastropods. It stumbles elsewhere, which suggests it's capturing a real signal, but not the whole signal. Imagine building a cathedral and discovering that the blueprints only explain half the arches. The other half stand on something you haven't mapped yet. Here's the part that most videos skip. Most deep-sea life is small. If you grabbed a random sample from 2,000 meters, the majority of what you'd find would fit on a fingernail. The giants, the ones that get the thumbnails and the documentary close-ups, are statistical outliers. The myofauna, organisms smaller than a millimeter, actually get smaller with depth. Not bigger, smaller. The pattern that Moseley described isn't universal. It's selective. Gigantism occurs in specific lineages: crustaceans, cephalopods, cnidarians, certain eels, and doesn't occur in others. Whatever mechanism drives it, it doesn't act like a rising tide lifting all boats. It acts like a permission slip. Certain body plans, under certain conditions, are allowed to scale up. Others aren't, and nobody has identified the threshold. Craig McLean, a deep-sea biologist at the University of Louisiana, studies exactly this paradox. His work shows that body size in the deep sea follows multiple overlapping gradients. temperature, depth, food supply, oxygen, and that no single variable predicts size across all taxa. If you're waiting for the clean answer, the one sentence that explains everything, it doesn't exist. The answer isn't one rule. It's a stack of rules, each one dominant in a different lineage, at a different depth, under different conditions. The deep ocean doesn't have one solution to size. It has several, running in parallel, sometimes reinforcing each other, sometimes contradicting. Like an orchestra where every section is playing a different score and somehow the result still sounds coherent, but only from a distance. And then there's the timeline. The genome of Bathynomus jamesi, a giant isopod cousin, was sequenced in 2022. The results were strange. The isopod's genome is enormous, packed with transposable elements, what geneticists call jumping genes. These sequences move around the genome, creating mutations at a rate far higher than normal. The researchers also found specific modifications to genes controlling thyroid function, insulin signaling, and fat metabolism, all pointing toward a biological program tuned for growth, storage, and extreme fasting. The isopod didn't just end up big. It's engineered to be big, down to the firmware. Evolution didn't passively allow gigantism. It actively selected for it, with genetic architecture that reads like a spec sheet for surviving in a place where food arrives once, maybe twice a year. And then nothing. For years. Which brings us to the question nobody wants to hear. What happens to the giants when the ocean warms? Current climate models predict rising deep-sea temperatures and falling dissolved oxygen levels over the next century. If any of the four theories are correct, Bergmann, Kleiber, island rule, or Chappelle-Peck, then the conditions that permit gigantism are shrinking. The deep ocean's thermohaline circulation, which delivers cold, oxygenated water to the abyss, is already slowing. The conveyor belt that keeps the deep sea hospitable to large life is losing momentum. And the giants, the ones with the lowest metabolic rates, the longest generation times, the slowest capacity to adapt, are the most vulnerable. Not because they're weak, because they're optimized for a world that might not exist in 100 years. You don't notice when the biggest things start disappearing. Not down there. Not in the dark. Moseley pulled a giant sea spider out of the Atlantic in 1873 and wrote it down. Chappelle and Peck measured 1,800 species and found a pattern. McLean mapped the contradictions. And the genome of a creature that hasn't eaten in 5 years told us something we weren't expecting. The deep sea doesn't just allow giants, it builds them. On purpose. With tools we're only beginning to read, the biggest things in the deep ocean are also the least understood. And one of them, the largest invertebrate predator on Earth, has never been captured alive. Everything we know about it comes from dead specimens pulled from trawl nets and sperm whale stomachs. Every diagram, every measurement, every model of its behavior, reconstructed from a corpse. That's next. And that, all of that, is what sits in the HADAL dark right now, doing things we can only guess at. Fact delivered. No invoice.